pollen nutrition structures, and bee and plant community interactions. Today, our group will present key findings from a recent study by Vaudo et al. On the role of pollen nutrition in shaping interactions between wild bees and flowering plants. While there's a consensus on nutrition being a critical factor for addressing pollinator decline, science has not delved deeply into the plant's nutritional content for wild bees. But as pollen is bees' primary nutrient, studying its macronutrient makeup in various plants can provide insights into how they might influence the bee populations that forage on them. This research highlights pollen's critical importance as a nutritional resource for bees, focusing specifically on the macronutrients, proteins, and lipids that are essential for their development and health. The study investigates how variations in the nutritional content of pollen from 109 co-flowering plant species influence the visitation patterns of 75 subgenera of pollen-collecting bees in the Great Basin and Eastern Sierra regions of the USA. One of the main findings is that the nutritional profiles of pollen can predict the composition of bee communities visiting those plants, suggesting that different bee species have specific foraging preferences based on the protein to lipid ratios in pollen. Furthermore, the research emphasizes the need for nutritional diversity in plant communities to support a wide range of bee species, which has important implications for conservation and habitat restoration efforts. As we delve deeper into the study, we will explore the specific macronutrients examined, their significance for bee health, and how this knowledge can inform our approaches to supporting pollinator populations. The image on the left displays a visual of bee foraging patterns based on the geometric framework method, which observes bees' nutritional needs and the composition of available resources in the Great Basin landscape. Essentially, what it shows is 3 distinct nutritional niches, which we'll get into more later. For now, I'll contextualize what the authors hope to find and their hypotheses. There were 3 research questions. First, how likely are plants with nutritionally similar pollens to be visited by similar bee communities? Second, what role does pollen nutrition play in structuring broad patterns of bee-flower interaction networks? And lastly, do plants with different pollen nutritional profiles differ in their levels of specialization or generalization within interaction networks? The authors hypothesized that plants with similar pollen nutrition attract similar bees, creating nutritional niches where specific groups of bees and plants interact frequently, and pollen nutrition shapes visitation patterns, with some plants acting as generalists, attracting many bee species, and others as specialists, attracting only a few specific types.
So you might be wondering why we care about bee nutrition. It's important for bees to get a mix of both protein and fat to stay healthy, and no single plant can meet the needs of every bee. That's why planting a variety of plants with different nutritional value is important when restoring bee habitat. Especially by studying which plant provides the right nutrition, we can create better habitats to support diverse bee populations and help prevent pollinator decline. Currently, scientists have studied more on how flowers attract pollinators using traits like color and shape, but they have not focused much on the actual nutrition bees get from pollen. Some earlier studies have been done with bumblebees and honeybees have actually shown that they prefer certain ratios of protein to fat, which is called PL ratios, to stay healthy and grow. However, a gap in these earlier research is that they are done in laboratories with only a few types of bees, focusing on controlled setting. For example, studies with bombus and patent showed how specific BL ratios affect their health and colony size, but these studies then look at how things work in nature with many different other bees. Thus, this research covers many more species, specifically 75 bee subspecies and 109 plants, giving a bigger picture of how nutrition affects bee-plant interaction in the real world. Additionally, it uses new ideas like nutritional geometry to show how bees pick plants with different nutrition to balance their diet. The study also uses a tool called SPV lipid assay to measure fat and pollen more accurately. Researchers match pollen nutrition with flower shape to find patterns more easily. They also made maps to show how certain bees and plants fit together based on their nutrition they need and provide.
Moving on to specific methodologies used in this study, I'll begin with how researchers observed and collect data on bees. The study was conducted across 8 sites in the Great Basin and Eastern Sierra region, spanning elevations from 1,400 to 2,850 meters. Observations occurred between April and August 2020, covering 66 days of data collection. At each site, researchers walked 4 to 10 miles along trails, service roads, and open meadows to locate flower patches with at least 25 square meters in size, with a minimum of 10 blooming plants. Once focal plant patches were identified, researchers spent 10 minutes observing each plant species per collection day, focusing on female bees that were actively contacting flower antlers. Additionally, bees were identified to the lowest taxonomical level possible, often down to subgenus levels. However, there were 2 species that were excluded from the study: Hylia bees and bumblebees. Next, let's discuss how flowers are categorized in this study. The study observed bee visitations across 138 wildflower species, where 131 of them were native. Flowers are categorized by shape and for architecture and symmetry to better understand how these traits influence bee foraging behavior. For flower shape and for architecture, flowers are categorized into 3 types: open, tubular, and complex. For open flowers like dandelions, anthers are fully exposed, making pollen more easily accessible. For tubular, they are recessed, requiring bees to forage deeper. And for complex flowers, flower shape is more complicated and pollen is hidden, needing specialized foraging techniques. Next, symmetry. There are 2 types used in the study: radial and bilateral. For radial symmetry, there are multiple planes of symmetry, which allow easier pollen access from any angle. In bilateral, there is only a single plane of symmetry, requiring bees to position themselves specifically to collect pollen. Lastly, researchers also measure floral sizes for each species, including average width and depth, to explore how size influences bee visitation and pollen collection. Finally, the study analyzed pollen nutritional content for 109 plant species, focusing on protein and lipid concentrations. Researchers harvested at least 10 milligrams of pollen per species using techniques like direct collection from flowers or extracting it from the anthers. Protein concentration was measured using the Bradford assay with an NaOH buffer, which extracted proteins over 24 hours and disrupted about 78% of pollen grains. Lipid content was analyzed with the SPV method, which used a chloroform, methanol, and water mixture and emphasized digestible unsaturated fatty acids like omega-3, omega-6, and omega-9. Finally, protein to lipid ratios were calculated to capture the nutritional balance that bees rely on while foraging.
Moving into the results, I will be addressing the findings based on the 3 research questions introduced earlier. So the first question focuses on how similar pollen nutrition influences bee visitation patterns. The first main finding is that plants with similar protein to lipid ratios were more likely to be visited by similar bee communities. This is shown by principal component 1, which explains 22% of variance between plant species based on the types of bees visiting them, and ultimately found that the connection between plants and bees is fairly strong with a slope of 0.52. This demonstrates that bees tend to choose plants based on how nutritious the pollen is, specifically favoring plants with a certain balance of protein and lipids. The second main finding is that bee visitation patterns were influenced more by pollen nutrition than floral morphology. The principal component 2, which explains 14% of variance, highlights nutritional impact with focuses on flower shape and symmetry. For flowers with a symmetrical shape, the exact balance of protein and lipids becomes more important for bees. For example, for bilaterally symmetrical flowers, bees are especially influenced by the pollen's P/L ratio with a slope of negative 0.58, and a higher P/L ratio resulted in fewer types of bees visiting these flowers. Next, the second question explores how pollen nutrition structures broad patterns of bee-flower interaction. Plant-bee interactions naturally form distinct modules or nutritional niches where plants share similar P/L ratio. This is shown in Figure A, where bee subgenera are grouped with plant genera that have similar P/L profiles. The darker color of the square represents a higher frequency of interactions between the two. These modules are associated with specific average P/L ratios, representing nutritional niches that guide bee foraging behaviors. Plants with high P/L ratios tend to attract diverse bee communities, especially those requiring high-protein diets. In contrast, plants with lower P/L ratio cater to bees that focus on lipids and show more narrow interaction patterns. These module groups consistently appeared through multiple analysis attempts, which validates the concept of nutritional niches and reflecting a reliable relationship between pollen nutrition and bee foraging behavior. Lastly, the final question examines whether pollen nutrition indicates levels of specialization or generalization within interaction networks. The figure here on the left illustrates the relationship between plant species network indices and their pollen nutritional value, which ultimately demonstrated that there are no strong trends across the different metrics. Plants with different P to L ratios are equally likely to have high or low indices, although there is a weak trend of higher pollen to lipid ratio plants being more specialized within bilaterally symmetrical flowers. So what this means is that plant species with varying P to L ratios show no strong trends in attracting either specialist or generalist bees. While pollen nutrition might slightly influence certain patterns, lack of clear correlation suggests that bee foraging behavior is influenced by factors beyond just pollen nutrition.
In conclusion, the key findings from the article were: first, plants with similar protein to lipid PL ratios were visited by similar types of bees. This showed that pollen nutrition actually plays a big role in shaping which bees visit which plants. This also means that we can use this knowledge to better manage pollinators and pollination and help protect bee population and biodiversity. Second, pollen nutrition had a bigger impact on bee visitations than things like flower shape or plant types. This means that focusing on the nutrition in pollen is more important when trying to support bee communities. Third, bee-plant interactions naturally form groups or nutritional niches based on PL ratios. These groups show that plants with different PL ratios attract different types of bees. This can also help with restoration of efforts by planting a variety of plants to support a wide range of bees. Lastly, pollen nutrition didn't strongly affect which bees were specialists, visited by only certain plants, or generalists, visited by many plants. Other things like habitat and competition also plays a role in how bees choose plants. In summary, pollen nutrition is very important for understanding and managing bee behavior. By using this information and looking at other factors too, we can better support bees and protect biodiversity.